Hello, everyone, and welcome. My name is Reagan Davenport, and I manage the teacher community of practice for the Amgen Biotech experience in the program office. Today, we're diving into the Amgen Biotech Experience Exploring Precision Medicine Curriculum. This material has significant relevance to both secondary and post-secondary education, and we're excited to show you exactly how it can support your students and programs. To guide us through this, I'm joined by two experts from our ABE implementation team. First, we have Tara Bristow. Tara, tell us a bit about your role and how long you've been with ABE.
So I have been with ABE since 2010, so about 15 years now, and I am the director of implementation. So I am assisting a team of technicians to make sure that things run smoothly at sites and to assist in developing new materials.
Thank you so much for sharing, Tara. Also with us is Susan Glick. Susan, please share your position and your time working on the ABE team.
Thanks, Reagan. I'm a senior production developer with the program office, and I've been working with ABE for a little over a year now.
That's great. And thank you both for those introductions. Again, we're excited to have you here, and I'm going to turn it over to Tara and Susan to get started.
So today we're going to walk you through the new version of the Exploring Precision Medicine Curriculum module. The first thing we're going to do is provide a brief overview of the curriculum. Then we're going to talk about the ways that the lessons are organized, including some different options for teaching. I'm going to walk you through the various chapters of Exploring Precision Medicine with this big picture infographic, which you can also see in your teacher guide. And we're going to start in the bottom left-hand corner with Chapter 1, Medical Mystery. And in this chapter, students are going to be introduced to a fictitious patient case of a patient who's having difficulty metabolizing a life-saving medication. And from there, they're going to contemplate any genetic differences they may have, which might have an effect upon their phenotypes for particular genes, as they consider in Chapter 2, thinking about whether they like particular bitter foods or not, and whether they are capable of tasting a bitter chemical called PTC. From there, in Chapter 3, where it's applicable, students will be extracting their own DNA from cheek cells. In places where this is not an option, we will provide template DNA. And they're going to use PCR to actually amplify a portion of one particular gene responsible for bitter taste. Then, in Chapter 4, students are going to use bioinformatics software and DNA analytics software to align DNA sequences, look for differences between those sequences, and explore restriction enzymes. In Chapter 5, students will return to the bench where they're going to be carrying out a restriction enzyme digest of some DNA that they amplified. And in Chapter 6, they will get to visualize their own restriction digested PCR products. They will finish up in Chapter 7 by investigating the genetics of drug metabolism.
So the EPM curriculum that Susan just described is divided into 7 chapters. The chapters highlighted in white are computer and discussion-based chapters that we commonly refer to as dry labs, while those in yellow are the hands-on laboratory-based activities that we commonly refer to as wet labs. The 3 wet labs take students through the process of determining their tasting phenotype, then extracting their cheek cell DNA, running polymerase chain reaction, restriction enzyme digest, and then gel electrophoresis. So these laboratories are a great complement to the Foundations of Biotech series and allow students to reinforce the skills learned. The 4 computer or discussion-based dry labs include exploring bioethics of precision medicine, data analysis, and sequence alignment. There are accompanying slides for each chapter and QR codes throughout the guides that link to the resource page where you can find videos, articles, and the DNA sequence files. There are also examples of various timeline options for lesson planning, which we will take a look at now. So the first option for completing the Exploring Precision Medicine curriculum is the full sequence timeline. And this includes doing all of the activities and would take about 15 days or 3 weeks for a class, which meets for about 50 minutes. We also have a fastest sequence timeline. If you want to do the entire sequence of 7 chapters, but don't have a full 3 weeks, you could still accomplish this in about 11 days. It does involve assigning certain portions of the dry labs as homework. So the third option is a lab-only timeline. And for this particular one, you would just be doing those wet labs that were described. And you can implement the 3 relevant chapters in about 5 days. So if you choose the lab-only option, there are 1-page descriptions with accompanying flowcharts that could be used in place of printing the entire curriculum. These can be placed into protective sleeves and put on your bench tops and make it a little easier for your students to go through the activities.
So now that we've given you a sense of the curriculum, let's take a closer look at how we've implemented the so-called 5E science education model in these activities. So you'll notice in this example from chapter 4 that we focus on the engage, explore, and explain components of the 5E model, leaving you to come up with your own extensions and evaluation as necessary. So why are we looking at PTC and the ability to taste bitter substances? For one thing, it's of relatively little medical importance. And for another thing, everybody has opinions about food. And so this is a good engagement point to get students interested in looking at precision medicine through the lens of something that affects them personally. The first thing you can do is to ask students their opinion of foods like raw broccoli or bok choy or coffee or other bitter foods. We also have an option. We have a list of cross-culturally bitter substances for them to think about. Why is it that some people like these foods and others can't? There is a genetic component. It turns out that people have naturally occurring genetic variation in one particular bitter taste receptor, a gene called TAS2R38, which makes a protein that's expressed in the taste buds. Let's take a closer look at the molecular biology of taste. Each of the so-called 5 tastes has its own receptor cell located in the taste buds of your tongue: bitter, sour, salty, sweet, and umami. And each taste receptor cell has its own receptor proteins. So when a taste chemical in your food binds to one of these taste receptors, the receptor cells send signals through gustatory neurons to your brain, and you experience the particular taste. The gene that students are looking at is the taster receptor 2 member 38 gene, one of an entire class of bitter taste receptors. How does the experience of bitter taste work on a molecular level? You need to have a taster allele, at least one copy. And the so-called wild type condition, which is the one that's found most frequently, is for people to be tasters of PTC. And there are combinations of variations of that. There are 2 alleles. If you're a taster, PTC binds to the receptor protein, which is located in the cell membrane of the taster cells. And if you're a non-taster, PTC can't bind to that receptor. If PTC binds to the receptor, it sets off a molecular signaling cascade, which results in a signal being sent to neurons and ends up in the brain. Students are going to learn about the interplay between phenotype and genotype, as well as some key molecular biological concepts when they amplify and carry out restriction digest on their own taster DNA. Students already are aware of their taster phenotype because they've carried out the PTC taste test. There are a range of experiences they could have. If the PTC tastes absolutely terrible, chances are good that they are homozygous tasters or so-called strong tasters. If it tastes kind of meh, but it's definitely yucky, they are most likely weak tasters, in which case they would be heterozygous with one taster allele and one non-taster allele. And if it tastes like nothing but paper, they are non-tasters. they are homozygous for 2 non-taster alleles. It's important to emphasize in each case that the students have inherited 1 allele from each parent. And if we represent the taster genotypes with letter T, we've got strong taster would be homozygous capital T, weak taster the combination, and non-taster homozygous lowercase t. The next step would be to determine, is there a genetic test that allows students to differentiate from on a molecular basis between their different potential genotypes? And the answer is that there is a restriction enzyme which can make a cut that differentiates taster and non-taster DNA. To be more specific, HEY3 cuts taster DNA and it does not cut non-taster DNA. So students will have amplified a 240 base pair fragment. And once they subject this PCR product to restriction enzyme digestion, if they are tasters, they will end up with 2 bands. If they're non-tasters, they'll end up with a single band. And if they are heterozygous, they should see a combination of all 3 bands.
You may be asking why you should consider implementing the Exploring Precision Medicine module. There are a few key considerations. So number 1, the EPM curriculum itself is very flexible and can be implemented in a variety of different ways to meet the needs of your classes. The resources, such as the slide deck, can be easily adapted to meet the needs of your student audience. Secondly, PTC Tasting Lab allows for exploration of relationship between the phenotype and genotype in the context of something that can help students understand medically relevant traits. And lastly, the discussion-based and bioinformatics activities allow students to analyze data and align sequences.
Thank you, Tara. Thank you so much. So we would like to thank everyone for watching this webinar. We want to let you know that this is the first episode and that we will have 8 more episodes with a mix of pre-recorded and live sessions available throughout May 2026. And so our next episode will be Bioethics in Precision Medicine, followed by Understanding Lab Contexts, Laboratory Implementation, Computer-Based Labs, Supplementary Resources for Educators, Best Practices for Implementation, Career Trends in Precision Medicine, and then we will wrap up by providing some extensions for the Exploring Precision Medicine Curriculum. The dates of those webinars are listed as well, and that is when you can expect to see a link that lets you know that the episode is available on YouTube. For webinars that we'll be recording live, we will send out an email with information on how to register. We would also like to provide you with a QR code for the Exploring Precision Medicine resources that you can scan to access the resources and to download the PDFs. We hope that you've enjoyed this overview of Exploring Precision Medicine today, and we'll see you for episode 2.